My name is Sally, and it's my pleasure to introduce today's speaker, Dr. José Santos. Dr. Santos is a veterinarian with postgraduate research and clinical training in ruminant nutrition, reproductive physiology, and dairy production medicine. His primary research efforts focus on the interface between nutrition and reproduction and methods to improve postpartum health and fertility of dairy cows. Dr. Santos received his Doctor of Veterinary Medicine from Sao Paulo State University in Brazil. He completed his Master of Science and his PhD at the University of Arizona with a focus on ruminant nutrition. He completed his residency at UC Davis with a focus on dairy production medicine. Dr. Santos is currently a research foundation professor in the Department of Animal Sciences at the University of Florida, and he is an adjunct professor in the Department of Animal Sciences at Sao Paulo State University in Brazil. In today's seminar, Dr. Santos will present his work on integrating energy balance and fatty acids to improve reproductive processes in dairy cows. Please join me in welcoming Dr. Santos.
Well, thank you very much for the opportunity to come and visit. It's indeed a pleasure. I had been to your campus here a few times before, but the last few days I had the opportunity to sit down with people that I truly admire, Dr. Baser and Dr. Wu and others here in the faculty, and discuss science, and hopefully there will be future collaboration. So I'm very, very grateful for this opportunity here. So when I received the invitation, I knew that there was probably different interests in terms of people with maybe more emphasis in reproductive biology and people with more emphasis in nutrition. So I start thinking about what should I put together relative to what I do. And so I came up with this title, and hopefully I'll be able to develop some of these concepts with the animal species that I really like to work with, which is the dairy cow. So I'm just going to give you some historical perspective. This is the cow that we had in the 1940s, so 70 some years ago. And this is a graph that you will see. This is the milk production per day. This is plotted on a weekly basis. And this is the estimated milk yield for a cow that's producing about 4,500 pounds of milk, which was the average production in the U.S. at that time. And this would be the potential intake of that cow in terms of dry matters. So you see here the relationship. This is not obviously a caloric or nutrient relationship, but it's simply a kilogram relationship that this cow here, because of her genetic makeup, she was able to consume in terms of kilos very similar amounts relative to the kilos of milk that she was secreting on a daily basis. So very little challenge as long as she was provided with resources, which wasn't always the case, and that was the challenge then. It's quite different today. I argue with colleagues in genetics. They think that genetics is 90% and environment is 10%. I'd like to think it's a little bit different than that, but that's how it goes. So obviously genetic selection has accelerated the process, and we really changed many aspects in the biology of the cow. One of them is the mammary gland, either the secretory activity, the shear size, and the ability to extract nutrients and put that into milk. So here now you have the average cow in the United States that in 2015 produced 10,500 kilograms of milk. So a little over almost 23,000 pounds of milk. And you can see now, at least in kilograms, we see a large gap here in terms of kilos of milk secreted in kilograms of intake potential of that cow. And obviously, when we talk about the average cow, there are cows today, as you will see, are well above the average. And I'll make some parallels to other animals, or at least with humans. So this creates a period of insufficient nutrient intake, not just calories, but any nutrient that you can think of. I can make a case that in early lactation, the cow will draw on to reserves. So here I put two extremes. This would be today the genetic potential of the dairy cow. This is a cow from Wisconsin that produced almost 34,000 kilograms of milk in a year. She was just a regular cow that actually purchased by the producer, and he raised that cow after he purchased her as a heifer. So it's amazing, and you will see what this means in terms of kilograms of amino acids and calories. And this is the world record in terms of lifetime achievement. This cow produced a 250,000 kilograms of milk over 15 years in 10 lactations. It would be the same as a cow milking no stop from today until the almost 4,500 days, no stop, producing 50 kilograms of milk, which has very high quality and concentration of nutrients. So there is a third cow that's not listed there, which is producing even more than that. Okay, but because she was not verified, so she's dropped from the world records in terms of lifetime or single lactation achievement. So obviously this puts a major strain in terms of regulating body nutrient supply and partition of nutrients. So just to put this in perspective, I read a few papers before I came here to look at some of the epidemiological literature of how top performing humans do. I don't necessarily work on this area, but I trust the colleagues that I read. And this is a survey from one of the Olympic Games, US athletes. Obviously, you have high caloric demand athletes and you have less caloric demand athletes, and that's why the range. So they are consuming 2,300 kilocalories or 2.3 megacalories per day up to almost 5 megacalories per day. Lance Armstrong, for example, during intensive training, your fellow Texan here, he is consuming 6,000 kilocalories per day to maintain pretty slim body size. Yeah. So the top performing human, to put in perspective, is consuming about 1.2 to 3.2 times the maintenance requirement if you were to assume that the average person needs 1,900 kilocalories per day. Okay. So if we look at some of the top performing cows and put that in perspective, so here is a former world record, and you can see she's not an exceptional cow in terms of size or beauty or anything, but here is her actual lactation curve, and the scale here is in kilograms. So if you were to translate at peak milk yield here, this cow was producing 220 pounds of milk per day, which is quite phenomenal in terms of nutrient extraction and packaging. And if you put this in perspective, that's equivalent for those who work on beef cattle nutrition, for instance. In terms of calorie, 15 kilograms of body weight deposition per day, if that was possible. In terms of metabolizable protein supply, about 21 kilograms of body weight. Or in terms of fatty acid supply, this cow is consuming about 3.4 kilograms of fatty acids, and she's secreting approximately 100 grams of linoleic acid in terms of one of the essential fatty acids. So what does that mean relative to her maintenance needs? Okay, she has to consume nutrients at almost 9 times her maintenance needs. Obviously, she's a superstar of the cows, but if you look at the average dairy cow, that's the average. Any cow out there, when she gets to about 50 days in milk, somewhere here at peak lactation, she's consuming 4.5 to 5 times maintenance. So you can see that this average animal that's been designed to be highly productive, if things go wrong, nutrient supplies alter, many bad things can potentially happen, or at least productivity can be impaired. And I'm going to drill on that a little bit during the discussion. So from a conceptual point of view, I'm interested in reproduction as well. And we know that when we think of species, obviously reproduction is essential for the survival of the species. But if you read the wildlife literature, it's very easy to demonstrate that once resources are scarce, the individual animal will have major impairment in reproduction because reproduction is a threat to the individual. And obviously, there is a priority on how nutrients are utilized. If we think that cells require oxidizable fuels, during plenty, we deposit those nutrients in storage sites such as the muscle and the liver or adipose tissue. And when we need, we basically mobilize that to supply to the tissues that have the highest priority, such as those that are in charge of essential processes, such as pumps, ion transport, neural communication, muscle activity. So those things are going to be the priority. And then we have processes that are called reducible, such as maintenance of body temperature, locomotion, and obviously in the world that I live, lactation is going to be modulated by nutrient supply. But lastly, for those of us who actually like reproduction, we are at the bottom of the scale when it comes down to how nutrients are partitioned, because obviously pregnancy during periods of scarcity presents a challenge to the dam, and there has to be some evolutionary mechanisms to control reproductive processes when the environment's not adequate from the metabolic point of view. And that's why there's a lot of communication between the splenic tissue and the central nervous system controlling reproductive processes. Now, I'm going to show you just one piece of data, because probably most of the data in bovine has been generated in beef cattle here at this department. But in dairy cows, one of the linking mechanisms that communicates the gut or the splenic tissue with the brain and reproductive process, or the gonads, is the liver and the IGF-1 growth hormone system. Okay, so here's a study from Cornell University in which they induce negative energy balance in cows, basically feed restriction, with the idea that they would uncouple the growth hormone IGF-1 axis. And then they came back and they replenished insulin as being the recoupling mechanism or the missing link in the individuals that didn't consume sufficient nutrients. Okay? And here is the mRNA for growth hormone receptor 1A. This is the most sensitive growth hormone receptor in the liver that actually is altered during nutrient supply. And you can see that there is an upregulation in the message for that particular gene, as well as IGF-1. And obviously, the liver is the main producer of IGF-1, at least in the cow, but probably in most mammals. And with the increase in insulin, which, if you were to put in the perspective of nutrient supply, in the case of the ruminant, it's dictated primarily by the provision of propionate in the rumen that gets converted into glucose during gluconeogenesis. But if I artificially create a hyperinsulinemic alglycemic clump, I can increase insulin despite the fact that I didn't improve nutrient supply, and that increases IGF-1 total and free IGF-1. And this recoupling mechanism is critical to stimulate resumption of ovarian cyclicity during early postpartum in cows. So we think that one of the links between caloric balance and reproductive process is mediated by the splenic tissue. One example is the recoupling of the growth hormone IGF-1 system. Okay, so I told you that cows produce a lot of milk. The extremes now at 34,000 kilograms in the lactation, the world record. And when people look at this, they say, well, milk is the reason why cows have poor reproduction, because there's a major drain of resources by doing that. So here is data from 160 cows that we have energy balance in the first month postpartum. So we have daily body weights, daily feed intake, the estimated caloric density of the diet, and then milk production and milk composition. And you can see that if you calculate energy balance as being energy consumed minus the maintenance needs and whatever other needs the animal might have, in this case lactation, which is the biggest player, there is a tight relationship between energy intake and energy balance. Okay, so you can explain a lot of the variance in energy balance relative to energy intake. But if you look at energy balance relative to the amount of energy secreting milk, that relationship is very weak. And if you look at energy balance relative to the maintenance needs of the cow, which is the body size in this case, or the body weight, that relationship is also weak. It doesn't mean that production doesn't affect energy balance, but it means that if I were to put effort and resources, I should probably pay attention more on energy intake or caloric intake than necessarily suppressing milk production to make strides in terms of improving energy balance in early lactation. And the reason why we're interested in nutrient balance is because if cows undergo massive negative energy balance, so let's say minus 20 megacalories of net energy in this case, that would be a cow losing about 4 kilograms of body weight, of which 90% comes from adipose tissue in terms of calories. So this is massive catabolism. That has major long-term implications to reproduction as well as other aspects of the cow. So today, when you talk about improvements in nutrient supply in early lactation with the goal of improving reproduction, the key message is how can we make cows consume more nutrients? The problem is that our understanding of control of food intake in dairy cattle is limited, and there's way too many external influence that affect that are very difficult to control, such that if you model intake of dairy cows, most of the variance goes to either the individual cow, the site, or to the residual of your model, and very little is, about 24% is explained by whatever fixed factors you have in your mathematical model that you know, some feature about the animal, how far in lactation she is, so on and so forth. So our understanding is limited. We have made some progress, but we still don't understand it very well. Now, having said that, industry has made major strides. So everybody knows that dairy cow has been an animal with reproductive problems. But I'm going to show you data from the US dairy industry. There's about 27,000 farms out of the 50,000 dairy farms in the US represented here. And I rank those farms as being very mediocre in reproduction, okay? Maybe some of you are not familiar with the metrics here, but this value here means every 3 weeks, every 21 days, which is the estimated length of the estrus cycle in the bovine, how many animals do I get pregnant out of the potential pregnancies that I can obtain? So given that the cow is cycling every 21 days, and she has passed the period that I want to start inseminating, of the potential pregnancies, how many a producer gets on his farm. And you can see some herds. The average in the United States is here now, 17% for the 9 million dairy cows in the industry. Some farms are really mediocre. Some farms are outstanding, okay? And you can see that this pregnancy rate goes up at the same rate that milk production has gone up. So I can make the case that there is a positive relationship between the two. It doesn't mean causality, although we've been bombarded with the idea that as milk yield has increased, reproduction has dropped. The producers are ingenious and they are smart and they use technology to overcome some of those weaknesses. So it is possible to achieve high milk production, 25,000 pounds of milk in a lactation with excellent reproductive performance. Okay? But cows face other challenges. And one of the challenges that is hard to account for is disease. Okay? So here is just to show you a very important disease, and then I'll relate this back of how this may affect reproduction in multiple facets, which is uterine disease that can cause massive inflammation in cows and cause fever. There's bacteremia in some cases. They show systemic signs, and it affects anywhere from 20 to 35% of the postpartum cows. And then if you look at other diseases that are not related to the reproductive tract, if you combine what happens here in this first 3 weeks with this first 21 days, which is also 3 weeks, so about 30 to 35% of the cows are actually affected by a clinical disease process. And 78% of all disease that happen in dairy farms, unless you have an infectious disease problem, they occur in the first 3 weeks postpartum. So this imposes another layer into that cow that's challenged in terms of nutrient supply. okay? Disease is not free, and I'm going to make a case that I can, based on another model, it's a beef cattle model, that disease can utilize a lot of nutrients. So a quick example is the synthesis of acute phase proteins, and there's a list of them there. And I'm going to show you one example here from beef cattle work, because I don't have a very good one from dairy cows that have nutrient flux across this plankton bed. So this is work from Oklahoma State University from Clint Kriebel. Unfortunately, it's a wonderful experiment, but I don't think it's been published still in a thesis, PhD thesis format, in which they had these Angus steers, 400 kilogram animals with catheters in the portal vein, in the hepatic artery, in the mesenteric veins, looking at the flow of nutrients and the extraction by the portal drain viscera and then by this plankton tissue as a whole, and then they target the liver. And I'm going to show you data just from the liver at this point, just so we can keep up with the presentation. So what they did is they took these Angus steers and they either fed restricted them for 72 hours or they fed them ad libitum. So they had enough nutrient supply, or they made them basically be anorexic temporarily. And then the model they were trying to study is respiratory disease, but I want to make a parallel to the cow that becomes very ill because of uterine disease that causes massive inflammation. And they put Mannheimia bacteria, the old Pastorella, which causes massive inflammation in the lungs. So they put bacteria in the lungs of some, so they challenged them or they didn't. So they had a factorial arrangement in their experiment, which was ad libitum intake, feed restriction, bacterial challenge, no challenge. And I'm going to focus on the bacterial challenge here. Okay, so here's just to show the acute phase response, aptoglobin concentrations in blood. And you can see that when you don't challenge the individuals, aptoglobin is at very low concentrations. Once bacteria is sensed by the sentinel cells in the respiratory tract, it sends cytokines that stimulate protein synthesis by the liver, and this protein has anti-inflammatory, antibacterial activity. And that increase is enhanced when the animal is off feed. So if I think of an animal that's ill, obviously it's going to develop inflammation, but it's also going to have sickness behavior, and sickness behavior involves depression and appetite. So here is the flux of amino acids across the liver. Okay? And in this case, basically separate in essential amino acids. So every time the numbers are below zero, it means that the liver is extracting more than it's secreting. So it's no surprise that the essential amino acids are under the negative values because they are not synthesized. But if you look at the control animals, the extraction of amino acids is less than in the challenge individuals. So if you look at the non-essential amino acids, now we see the opposite. The control have a positive flux. In other words, the liver is utilizing carbon and amino groups to synthesize particular amino acids and secreting that in the hepatic artery. Whereas the animals that were induced to develop inflammation, they were still consuming essential amino acids. So the end result is this differential net flux of amino acids. So how much is that for a 400 kilogram steer? That's when I calculate for the average molecular weight of the amino acids, because I don't have each of the amino acids listed there in this value. It's about 2.6 moles per day, which is about 380 grams of amino acids that would be secreted in the peripheral circulation. And if I put this in perspective for the animal that I'm interested on, usually the mammary gland has almost a 0.7% efficiency of extracting plasma amino acids to put into milk protein. And this is the equivalent of about 8 kilograms of milk in terms of amino acid extraction. So now you can easily see that if I'm interested in reproduction, the cow loses weight, but on top of that, 30-some% of them develop disease that imposes another nutrient need in terms of fighting infection and inflammation that adds to that negative energy status, which impairs even more reproduction. Okay, but disease has other aspects, and I'm going to show you one of them from work of a former grad student. So we collect day 5.5 to 6 more early blastocysts from single ovulating lactating dairy cows, and then we collect conceptuses from cows at a little bit later stage of gestation, day 15 to 16. And we look at different things such as whether we had cleavage, the quality, the morphological quality of the embryo based on the cell, the blastomere disposition, and then stain those embryos for cell number. And then in the later stage of gestation, we look at the length of the conceptus, interfrontal concentration in the initial flush, and then we did some transcriptome work in those. So here, so obviously this is not a experiment, so we can randomize cows to develop disease, unfortunately. This is a cohort study. Retrospectively, we basically look at, did she have disease, yes or no? We went to diagnosis in those cows before we synchronized them. And what we found was that the individuals that develop disease, they have less fertilization. So from the very early stages of gestation, they have reduced pregnancy just because we get less fertilization. When we look at the live embryos on day 6, smaller proportion. And if we look at the embryos that are classified as grades 1 and 2, which would be those that gives you the highest pregnancy, it's also depressed. When we look at that relative to the embryos only, so if we remove the unfertilized oocytes, that difference is retained. So we see a depression in pregnancy at very early stages, which is probably not a surprise. So if we look at the conceptus now on day 15, the elongation is impaired, so the length of the conceptus is less. And because we have fewer and less developed trifectoderm cells, we have less interfrontal in the uterine flush. So those cells are not secreting the typical proteins that you expect from at the same rate that you would expect compared to a conceptus that comes from a cow that did not develop disease. So obviously when you do microarray, you get massive amounts of data and massive pathways and networks. There were some that called our attention. One of them were those that were related either to inflammatory processes such as upregulation of seronamyloid A in cows, in embryos originated from cows that had disease, compared to embryos that originated from cows that did not have disease. So every time you see red, that particular transcript is upregulated in the conceptus of the cow that had disease, and these are the possible upstream regulators. So NF-kappa B, antigen-presenting cells, MHC, seronamyloid A. And you can see here that things that you would think of relative to inflammatory process are linked to that based on pathway analysis, okay, such as lipopolysaccharide, TNF-alpha, interferon gamma. When we look at the downstream effects of those genes from the pathway analysis in those cells, there is activation cells, there is systemic autoimmune syndrome. So our thinking from this, and obviously this is only hypothesis generating, is that inflammatory processes in the dam has a long-lasting effect that affects the conceptus, either because it affects the oocyte, because in the cow it takes 70, 80, 90 days from a primary oocyte to become a Graafian oocyte, and in a Graafian follicle that will be the ovulatory follicle eventually. Or maybe the uterine environment is altered such that the nourishment of that conceptus will be influenced, and it will express a phenotype that is imprinted with an inflammatory response, such that we may have maternal fetal interactions that can compromise pregnancy, such as presenting paternal antigens to the maternal immune system, for instance, through upregulation of MHC antigens in the conceptus. So we think that this may be linked to the reduced pregnancy that we see in cows, and I'll show you some data. Okay. When we look at interferon-stimulated genes, so that protein that was produced by the concepts that get into the maternal circulation and turns on genes in peripheral leukocytes, we see the expected response. The samples are collected on day 19, when we can't tell whether the cow is truly pregnant unless we kill her. Okay, we don't have any diagnostic tests for pregnancy that's accurate at that stage. But later on day 30, we pregnancy check that cow through ultrasonography. And the cows that are pregnant, they have a massive upregulation in the mRNA in the leukocytes for this one of the interferon-stimulated gene, in this case ISG15. When we look at the cows that had disease, our interpretation is that pregnancy wasn't quite substantial in terms of stimulating the turn on of the genes for interferon, in this case for ISG15, perhaps because the conceptus is less developed and there's less interferon tau getting to the maternal circulation. That's our interpretation from this. So we decided to go back and look at this and say, hey, can I demonstrate that the end product, which is a pregnancy, is altered? And can I bypass this if I transfer an embryo? And then hopefully if I transfer an embryo, I don't have the oocyte that was developed under that environment that may pose challenges to the cow. So I'm using now an in vivo or an in vitro oocyte. And that's what was done here. So here is pregnancy now on day 45. And we have cows here that have no disease diagnosed prior to the first postpartum insemination. There's about 5,000 cows here in this data set. And this is all first insemination. Some cows receive an embryo, some cows receive the AI. Okay. And we had cows that had non-uterine disease only. So they had mammary disease, respiratory disease, locomotion disease, so mastitis, pneumonia, so on. And you can see that pregnancy drops. Now we have uterine disease only, metritis, so massive inflammation or lactation. Okay, pregnancy drops. Now they had both. It drops even more. So we see this additive effect at this stage here, at least, that one adds to the other layer. When we look at pregnancy loss from day 45 to calving, we see twice the relative risk of pregnancy loss in animals that had more than one insult prior to the first insemination. This is not disease that's occurring after the cow is inseminated. It's actually a memory that's lasting a long time. Okay. So when we transfer an embryo to those cows, we are not able to bypass that lesion that disease has caused. Okay. You can see these are healthy cows that received AI. These are diseased cows that received AI. So we see a depression in pregnancy, an increase in pregnancy loss between day 45 and term. When embryos were transferring to healthy cows, pregnancy was higher than in diseased cows. The diseased cows, again, we had depressed pregnancy and an increase in pregnancy loss. Okay, so it seems that there is this memory that although pregnancy has been established, such as here, many of those pregnancies will not be carried to term, and the risk is greater if that cow had a previous blemish, such as disease. Obviously, disease causes inflammation, but one of the things that came out from the microarray work, as we look at concepts, there's obviously many genes that are altered during development. One of them was PPAR gamma, and there's work here from Texas and from Washington State showing that in the sheep, and then from other people in the rat, that PPAR gamma, which is a ligand for fatty acids, is very important during development, and there's massive upregulation in the gene, and if it's knocked out, pregnancy doesn't progress. A very interesting thing was that cows that had inflammatory disease, we had no expression in the microarray for PPAR gamma. So we think that these alterations are linked to the stage of development that's being altered, but also with changes in the cell, either mediated through inflammatory molecules or by other means that is affecting critical processes for embryonic survival or conceptive survival. Okay, so I'm going to try to make a link now with the nutritional approach trying to intervene on this. So I mentioned to you that PPARs are important. There's more than one of those, and PPARs are ligand for fatty acids, and fatty acids can have nuclear effects through PPARs. And usually those fatty acids that have major biological functions, they tend to be the more unsaturated fatty acids, at least it's better described in literature. And some of them were called essential because we're not able to synthesize, at least mammals were not able to synthesize. And these two scientists in Minnesota in the early 30s, was a couple, a husband and a wife. They did some very interesting work in guinea pigs and in rats, demonstrating the essentiality of two of those fatty acids, linoleic acid and alpha-linolenic acid, two different families, one an omega-6, the other one an omega-3 family fatty acids. Okay, so as with humans or other mammals, ruminants in intensive feeding systems, they consume diets that are very high in omega-6 because cattle are fed corn. So in general, plant products, with a few exceptions, canola and linseed or flaxseed, they're very high in omega-6 and very low in omega-3. And as we move from grazing systems into more intensive systems, N6 fatty acids became the prevalent, or the main source of unsaturated fatty acids in cattle diets, and very little N3. And obviously they have quite distinct functions in terms of cell biology and quite distinct activities in terms of stimulating or suppressing particular gene pathways in the cell. Okay, so here's just to show an example. Here would be the acyl chain of an 18-carbon fatty acid with a carboxylic group and methyl group. Usually, there's enzymes that can add double bonds to saturate that acyl chain, but in general, mammals only express genes for the delta 5 through the delta 9, and in general, there is no other desaturates beyond carbon 9 that's expressed in mammalian cells. Whereas plants can desaturate fatty acids up to carbon 15, and lower plants such as moss or lichens can desaturate even more, and then insects can produce a wider range of polyunsaturated fatty acids. Meaning that if I have a omega-3 fatty acid, in the case of 18-carbon fatty acid, so here would be my methyl group, and omega-3 would be double bond 3 carbons away from the methyl group, I have to consume that from some source in the diet, either from other products, other plants, or other animals that consume other plants, which is typically what happens in the case of fish fatty acids that eat algae in the ocean. Okay, so the challenge for us is that the rumen alters everything. Okay, in a monogastric, you feed X, hopefully X shows up there and gets diluted with tissue secretions and other things more. In addition to that. But the microflora of the pregastric fermentation doesn't like polyunsaturated fatty acids. They're quite toxic if they get incorporated into the membrane of rumen bacteria. So obviously bacteria evolve and they develop systems to alter the composition of those fatty acids. And they usually involve reductive reactions, such as in this case, from a linolenic acid, a 3 double bond fatty acid going all the way to a saturated fatty acid through a series of reactions in terms of isomerization, where you change the configuration of the double bond in the acyl chain, to hydrogenation reactions. So the importance of this is obvious that if I feed this, I may not get as much of this. That's one point. The second one is that during the process of biohydrogenation, there's massive production of numerous intermediates depending upon the conditions in the rumen. And some of them are very powerful agents in terms of biological effects. As a matter of fact, there is one fatty acid that is, as far as I know, is the most powerful anti-carcinogen, which is rumenic acid. It's a trans fatty acid produced only in the digestive tract of fermenting bacteria fermenting mammals. So rumen would be the main one, which is C18 to Cis9 trans 11. But the same way I produce some good fatty acids such as this here, there are others that can be produced that have other biological activities. For the dairy cow, the thing of major concern is that some of the fatty acids produced, and there are two of them, I'll show you some data, they have anti-lipogenic activity. Okay? So they depress gene expression for lipogenic enzymes, particularly de novo synthesis of fatty acid in the mammary gland. So the mammary gland doesn't take up as much substrate to synthesize short and medium chain fatty acids, but it also alters the activity of the enzymes. So not only you have less message, but you also less activity for the particular protein. Okay, so here just to illustrate, I may feed a cow and she's consuming 24 kilograms of diet. Ruminants get diets with low fatty acid content, relatively speaking, compared to monogastric, maybe 4%, maximum of 5.5% fatty acid. Okay? And this cow may be eating this many grams of stearic, oleic, linoleic, and alpha-linolenic. As it goes and travels through the digestive tract and microbial attack alters this, the amount that gets absorbed is completely different than what was fed. Okay? Part of that, there's some microbial synthesis, but primarily because of that biohydrogenation pathway. So we see massive declines in the unsaturated fatty acid fraction and a massive increase in the saturated fatty acid fraction. That's pretty typical of pregastric fermentation. This poses a major challenge for us if we want to study fatty acid nutrition and fatty acid biology, because we don't get the same 81 grams of oleic acid in intestine. Okay, so in terms of titrating those now becomes very difficult. It's really a trial and error to see what happens there. Nevertheless, we can show biological effects, and I'm going to show you some data. So we're very interested in the ones that we think have greater biological effects, such as the essential and the byproducts of the essential fatty acids or consumed in the diet, such as the polyunsaturated omega-3 fatty acids, as well as omega-6. I'm very interested in alpha-linolenic, DHA, and EPA. But obviously, in terms of reproduction, we can't forget others that may not necessarily be fed, but have major biological effects. So here's the situation in the dairy cow. This is flow through the digestive tract. These are grams of C18-2 linoleic acid, C18-3 alpha-linolenic. This is duodenal absorption and mammary secretion. This is a particular experiment, but I can show you 12 different studies that the picture is very similar. The study at University of California, Davis, cows were fed different fat supplements, and you can see that in many cases, the differential is minimum, yeah? In other words, the cow is secreting as much as she is absorbing in milk. In other words, there's very little left for any other function, such as replenishment of cell membrane, replenish of the unsaturated fatty acid pool, which in the cattle is primarily in the plasma pool. We don't really know what the requirements are for essential fatty acids in ruminants. And we use numbers from other species and we extrapolate. Okay, so I'm going to extrapolate here based on the rat data. And I'm using this value here for linoleic acid. So a 600 kilogram dairy cow, for her daily needs for maintenance, if she is a rat, which she is quite different than a rat, but if her biology is similar, she would require an additional 10 grams for her maintenance requirements to replenish whatever needs to be replenished. So during lactation, to the best of our knowledge, the supply usually doesn't meet the needs of the mammary gland plus the potential maintenance needs. But I have to say that if I undersupply, the mammary gland is very elastic and will secrete less. So probably what I think is happening is that the cow has vast amounts of lipids stored in the plasma as well as in the adipose tissue, and she's just using that, and at some point that's going to be replenished. It doesn't mean that they are not beneficial in terms of altering lactation and reproduction. I'm going to show you two experiments just to show some lactation data in which we fed cows diets that were very similar, in this case, in terms of energetic density. Across all treatments, they had the same protein content, fiber. And basically what we did is we had a control diet without supplemental fatty acid. This was the linoleic acid supply to those cows in terms of grams per day. Then we supplement primarily saturated fatty acids, okay? Stearic acid in the form of free fatty acid. So we increased fatty acid concentration in the diet, but we didn't change the polyunsaturated pool. Or we added calcium salts with an attempt to reduce biohydrogenation to some extent. It's not extremely effective, but we increase the intake of linoleic acid. So here is the milk yield and the fat cracked milk of those cows. This is the pre-treatment period in which they are in the same diet. And we can show changes in lactation performance by feeding diets that are very marginal in essential fatty acids and then supplementing them to the cow. And that happened in terms of milk, or if you were to put this on an energy basis in terms of fat cracked milk or energy cracked milk will be the same. Adding lipid to the diet stimulates milk yield, and adding additional essential fatty acids, in this case, that didn't suppress milk fat, it was able to alter lactation performance. And that wasn't at the expense of body reserves. Body weight was very similar across. Body condition score was very similar. There's a little bit less here body score, but small difference. So we think that, yes, there's a repartitioning of nutrients, but it's not at the expense of massive tissue mobilization. So I'll show you a second experiment now that we manipulate the supply of N6 to N3 fatty acids. So we move from a diet that had 6 parts of omega-6 to omega-3 to 4 parts of omega-6 to omega-3. Basically, we replace linoleic acid with fish oil fatty acids. And fish oil fatty acids are very toxic to rumen microbes. They can really destroy milk fat. So we made changes in the diet to accommodate for that, so we don't change the energetics of the cow because now she secretes less milk fat in the mammary gland. Okay, so it's a proof of concept type of experiment. And we did this by just altering the fatty acid supply to the cow. We maintain fatty acid concentration in the diets very similar. So the diets were very similar, okay, as you would expect. So here is the dry matter intake of the cows. We saw a tendency for an increase in intake, which is going to probably explain some of the milk response in those cows. This was the actual value after the analysis of the diets in terms of omega-6 to omega-3 fatty acids in the diet, a ratio of 3.9 to 5.9. So here is the consumption of total omega-6. It increases as I move to this direction. If I look at the total omega-3, it increases as I go to the left because I'm adding EPA and DHA to the diet. So these fatty acids here, we believe they have anti-inflammatory properties, and I'll show you an example. So again, the same type of response. We see as we increase the omega-3 now by supplying fatty acids that stimulate intake in this case, which was a surprise, actually improve fat cracked milk. We didn't cause milk fat suppression, so we didn't alter the energetics from that perspective, but the cows produce more milk, and we think there is a repartitioning of nutrients there because of their properties. Okay, I'm going to go back and forth now in a few things. This fatty acid can alter reproduction. So fat feeding in cows, there's a good body of data that shows improvement in reproduction. But the fatty acid per se can have differential effects. Here's one of them. So we basically fed cows diets that were low in linoleic acid or high linoleic acid. We inseminate them, and then we flush them 5 days later to look at fertilization, accessory spermatozoa attached to the zona pellucida, and embryo quality. And one of the things that we observed was that high linoleic acid in general improve those characteristics in the single ovulating dairy cow. So there's a case here that it may affect perhaps oocyte quality, oviductal environment, follicular composition, and there's some indication for that. But then in a previous experiment that we altered this ratio of omega-6 to omega-3, so we go back to that one, we collect endometrial tissue and conceptuses from those cows. And as you would expect, as we move from less omega-6, more omega-3 here, we see an increase in omega-3 fatty acids in the endometrial tissue. Okay, so here is one of them, EPA as percentage of the total fatty acid. It's usually accumulating the phospholipid component. And we see a decline in omega-6 fatty acids. Here's one of them, arachidonic acid, okay, as we increase the omega-3 fatty acids. Okay, so we see a decrease as we move from here to here, and we see an increase in the other one. That's how you would look at the reds and the blues. So they either is through competition or incorporation or availability, the cells are taking up those polyunsaturated fatty acids. In this particular experiment, we had the hypothesis that if we altered the endometrial fatty acid profile, we may modulate prostaglandin release spontaneously by the cow. And we were able to show some, but not striking difference. And here's just to illustrate, this is a progesterone profile in the cow, and this is right at the onset of luteolysis. We see some pre-luteolytic pulses of PGFM, and then the cow has big spikes of PGFM. And usually in the cow, as opposed to the sheep that seems to require 5 pulses, we can have very high sensitivity and specificity for CL regression if the cow has 2 large pulses. That's usually sufficient. And then that kills the corpus luteum and progesterone plummets. Here's a cow that didn't regress to CL. She has pulses of progesterone. So progesterone seems to be going up and down, large shifts in progesterone concentration every 2 hours. But she never had a signal from the endometrium to secrete sufficient PGF2 alpha, so PGFM is not high, the CL is not maintained. By altering the fatty acid makeup, we've been able to attenuate some, not sufficient to extend the interestrus interval. We had basically a day 0.2 increase in interestrus interval with the more omega-3, which we think is going to attenuate PGF2 alpha release, but not enough statistical power to show effects. But we were able to show that the pulses are attenuated relative to more omega-6 fatty acids. So altering basically the fatty acid makeup of the endometrium seems to affect PGF pulsatility, not to the point to alter interestrus interval. But yes, we can reduce the largest pulse during the luteolytic phase, as you can see here, more omega-6, more omega-3. So differences in the pulsatility of PGF, which we think that may be mediated by substrate availability, but also by changes in gene expression. So when we look at relative abundance for some of the genes related to the pathway of luteolysis, we see an increase in gene expression for estrogen receptor alpha, oxytocin receptor, prostaglandin synthase, as the amount of omega-6 fatty acid is increased. So there seems to be some gene regulation, which is not a surprise by those polyunsaturates, or maybe depression in gene expression by the fish fatty acid, fish polyunsaturated fatty acid, EPA and DHA. Okay, so these fatty acids are capable of altering membrane composition, modulating some gene expression, but it can also influence immune response in the cow. Here's data from Flavio Silvestre, former PhD student with Dr. Dacia, in which he collected neutrophils from cows that were fed either diets that were mostly saturated, monounsaturated fatty acids versus diets that were high in polyunsaturated fatty acids. And then he challenged those neutrophils when they came from cows fed mostly omega-6, safflower oil. And the omega-6 usually upregulates pro-inflammatory response, as you can see, production of TNF-alpha or interleukin-1 beta. When he shifts that, this is not in vitro, this is harvesting from the cows that were fed. So with all the biohydrogenation and everything that's happening, feeding fish oil fatty acids, so rich in EPA, DHA, was able to attenuate those responses, as you can see here. So yeah, we can change cell phenotype by altering the fatty acid supply to the cow. So here is a recent experiment that Leticia completed, and just to show you the challenge that we face. Okay, so here's the relative change in milk fatty acid. Algae is rich in DHA. This is what we use to deliver polyunsaturates in this particular study that we're looking at fertility, but obviously for us, milk yield and milk composition is still important. These are the fatty acids, up to 14 carbons, that are synthesized de novo through lipogenesis in the mammary gland. And they tend to be the ones that are depressed during what we call milk fat depression. And you can see that when we fed EPA and DHA, we depress the no synthesized fatty acids. So we're getting only 90% relative to the control diet. But we also increase particular isomers of conjugated linoleic acid. These two isomers have been shown to cause milk fat depression. So through the process of rumen biohydrogenation of linoleic acid, because EPA and DHA can alter the pathway by being toxic to some subgroups of microbes, we're able to increase the concentration of these two. And these two here suppress the enzymes that synthesize this. Okay? But we are able to increase EPA, we're able to increase the total N3 fatty acid, and we're able to massively increase DHA because our product was high in DHA. So we are providing about 12 grams per cow per day. So what happened when we fed this? I'm going to have maybe 3 or 4 more slides and I'll finish. This is interferon-stimulated genes. On day 19, we have open cows. We have pregnant cows that were fed the control diet or the DHA-rich diet. And you can see that for one of those genes, RTP4, we see upregulation of mRNA in the leukocytes. Those leukocytes, they have much greater expression when they come from primiparous than older cows. They're two different beasts in terms of that, but the response to the same. We see an increase in upregulation of that. So we think that this may be influencing embryonic development. What happened to pregnancy? We saw an increase in pregnancy after insemination for cows that were, so I know that this is kind of hard to see, but these are the primiparous. You can see the EPA supplemented animals had higher pregnancy. You can see for the multiparous, it's a little bit less response. So if you look at across over time during the lactation, the algae group or the EPA supplement became pregnant faster. This is a survival curve. You can see the hazard of pregnancy increased 38%. So basically we see stimulation in pregnancy. We think that's associated with embryonic survival. Let me show you the last slide, which is work from Flavio Silvestre, where he tests that idea. Prepartum and in early lactation, he fed one diet, which is this cold here. And then from 30 days postpartum until about 160 days, he fed a different fatty acid source. Okay? So he has mostly saturated prepartum, mostly saturated during the breeding period, mostly N6 saturated, most saturated in N3, N6 and N3. And you can see that the highest pregnancy was obtained when this combination was provided. And we think there's a reason for that. We probably don't have time to go into that. But one of the benefits was those N3 fatty acids, EPA and DHA, they reduce pregnancy loss. So we think that there is cellular communication that's attenuate perhaps inflammatory response, maybe altering antigen-presenting cells in the reproductive tract that's facilitating maintenance of pregnancy when particular fatty acids are provided. So just to summarize, dairy cows undergo extensive catabolism in early lactation. It's actually a phenomenal biology to study because the amount of weight loss, probably just marine mammals lose more weight in that particular period. or hibernating animals, yeah? And that delays estrocyclicity and compromise establishment and maintenance of pregnancy. On top of that, unfortunately, they have a lot of disease, and that adds another layer of complication. Supplementing fatty acids to dairy cattle diets influence energy partition fertility. Can be positive, but can also be negative if I cause major milk fat depression. Response to fat feeding in terms of reproduction, they are best when moderate amounts of unsaturated fatty acids of the N3 and N6 families are fed. There's always the fight between improving fertility but not affecting milk composition and milk energetics, so that's always a concern. Sources rich in N3 fatty acids seem to increase pregnancy by improving embryonic survival and altering some of those markers of inflammation, whereas sources rich in N6, they improve pregnancy because of increased fertilization and embryo quality, but there's many other effects on immune response, certain lactation that may mediate those benefits to fertility. So with that, I just want to thank a lot of the people who did all the work. Collaborators, Dr. Thatcher's been a great mentor all my career. Charlie Staples, who's a colleague now, we work together. The grad students who did all the work, Rafael, Eduardo, Leandro, Fabio, Natalia, Ibru, Leticia, Achilles now, and Gabrielle. Obviously the funding from USDA and private industry. And hopefully we'll have time for a few questions. Thank you very much.
We have time for a few questions. We're running a little late. Anybody have any questions? We'll bring that up. There we go.
José, what's known about fatty acid transport across the uterine?
There's at least two fatty acid transports that I'm familiar with. CD36 is one of them. And then you have fatty acid transport proteins. They are expressed in the placenta. They are expressed, obviously, in the small intestine and in the liver. There is preferential uptake. Usually, I'm not sure about the placenta, but the liver and even in the intestine, there's preferential uptake. Usually, the fatty acid the liver doesn't like is stearic acid, C18-0. There's very little uptake. In the placenta, there is massive uptake of polyunsaturated fatty acids, particularly arachidonic acid is one of them that gets incorporated in the endometrium. So arachidonic during certain stages of the cycle after progesterone stimulation gets up to almost, I think, 17 or almost 20% of the phospholipid fatty acid composition. Placenta, I can tell you how much gets incorporated just in the tissue and how much gets transferred to the fetus, but I would like to think that particularly because of nervous system development, some of these PUFAs are essential. So at least linoleic acid has to be taken up in large quantities because you have high concentration of arachidonic acid in the brain of infants, and there has to be preferential uptake for PUFAs as well.
Does the PUFAs supplementation increase the blood flow to the lactating fatty tissue?
It's a very good question. I don't know. Yeah, does PUFA, do any of the PUFA stimulate blood flow to the mammary gland? Fat per se has an effect on efficiency of nutrient extraction in the mammary gland. And one of the responses to fat feeding is a decrease in amino acid uptake by the gland because you meet the energy requirements for every kilogram of milk synthesized a little bit faster when you increase the lipid pool that gets into the mammary gland, either through free fatty acids or through HDL supply in the case of ruminants, which is the main, and chylomicrons. But people haven't really tested whether different fatty acids have modulatory activities in nutrient extraction, but feeding fat reduces the number of liters of blood that have to go through the mammary gland for every kilogram of milk. So it changes actually from about 900 liters to about 700 liters of blood going to the mammary gland in one particular experiment. But I can't tell whether different fatty acids do that. So the end result with feeding fat is that you decrease milk protein. So milk protein content goes down, although the cow produces more milk, so the total protein secretion is greater, but per kilogram of milk you get less amino acid extraction.
Okay, in that case, let's thank Dr. Santos.
Thank you very much.